Hey, good morning, ladies and gentlemen. Welcome to the show. It's good to see you in the audience. It's the first time we've done a morning show, but I'm really excited. Today's going to be an interesting show. We have Dr. James White from Rutgers University, who studies a lot of the stuff we're interested, like at the real fundamentals of a living soil, how plants interact with living soils. That's what he studies. Which, as a quick little background, a quick little story is when I was developing my Happy Endings tea, I wanted it to be really good for the roots. So I started asking fundamental questions like how does phosphorus or nitrogen get absorbed into a root? So I started looking around for some academia papers, and it turns out Rutgers University does a lot of that. And I ran into his work a number of years ago. And a couple weeks ago, I saw a presentation that Dr. White did that involved cannabis. So that really perked me up. Anyways, I'm going to bring him up here in just a second. But he did have a nice presentation to give us, but we're running into technical difficulties, so I'm not sure if we're going to be able to pull off the presentation. But I think it'll be just fine to have a talk with this gentleman, and also Wendy Kornberg, who's been on the show before, has volunteered to help me moderate this. So she's dropping her there. Up, actually, there she is, right there. Welcome, Wendy. I see you down in the green room. And at this point, I'm going to pull up. I'm done. You don't need to hear me anymore. Let's welcome Dr. James White. Good morning,
Hi.
sir. Or afternoon in your area, I guess, right?
It is. It is. It's 2:04 right now. Yeah, no, I'm happy to be here. Thank you, Patrick, and can't wait to meet Wendy.
Okay, let's meet Wendy. Let's go ahead and meet Wendy. Hi, Wendy.
Hi, Wendy.
Hi, how's it going?
Great.
Awesome,
Good.
great to have you, James. I'm super excited to hear your presentation, and I've got a bunch of questions. So,
Well, I don't know if I'm going to be able to show the presentation, but I mean, I could try and see if it'll work. It didn't work the first time earlier, but maybe it'll work this time. And actually, I sent Patrick a PDF of the presentation, so everything will work with that PDF. Except the movie files. Patrick, you can check and see if it's there.
I will do that. Wendy, yeah. You're going to take over for a minute, and let me check and see if I can get that PDF working. So,
all right.
Okay.
Let me, I'm a little bit of lag time, so let me actually check. Here we go. Let me get rid of Safari and see if that's a little better. And now it's totally frozen.
Although I can still. Why? What did you have? Because we can see you and hear you still.
Okay, perfect.
With no problem. Yeah. There's no problem at all. So, do you happen to have a PowerPoint on your computer? You have PowerPoint? What? Do you have PowerPoint on your computer?
I do have PowerPoint on my computer.
You do? Oh my goodness, because that might mean that I could send you that PowerPoint file, and you could, although I don't know your email, so I'm not going to be able to do that. So anyways, let's not worry.
Send you my email in the comments section on the chat if you'd like.
Yeah, send me that.
I'm just going to do it on private chat so it's easier to find just in case it doesn't get buried.
I see it.
And it's just wendy@sunibus.com, so it's pretty easy, but we'll send that away. There you go.
Ah, I see. Okay. Yeah. So I will just send you that. And let's see, send, and okay, here we go, and I'll go forward, and I'll give you this.
Okay.
And you should have this file directly, and I mean, we could actually start talking now.
Okay.
Like, yeah. So, okay. So, do you want me to introduce myself? You've
Go
already
ahead.
introduced me a little. Okay. So, I'm James White at Rutgers University, a professor here, and in the Department of Plant Biology. And what I study are endophytic microbes of plants, bacteria and fungi. Actually, I'm also a mycologist, so I teach fungi, mycology, mushrooms and fungi that cause diseases and so forth here at Rutgers. So, and I'm a botanist. I've been a botanist from the very early days of my career. Got a bachelor's in botany and a master's in plant pathology and a Ph.D. in botany. So I mean everything about botany. That's basically what I do, and with the specialization of microbes that go into plants. And over the past few years, we discovered that plants actually are internalizing into roots bacteria from the soil. They just take it, sucking those microbes right into the roots. And then they're using those microbes to stimulate growth of root cells and roots themselves, root branching, root architecture, as they call it. And they're also using those microbes to get nutrients, soil nutrients. They're using superoxide or reactive oxygen to oxidize those microbes and get nutrients out of the microbes. So it's interesting what plants are doing. And this is something we don't think of plants as actually eating microbes, but they are. They're feeding on these microbes. They're cultivating the microbes. They're attracting them to the root, and then they're taking them into the cells. They're eating them, so to speak. They're eating them, and then they're breaking some of them all the way down. Others they process, and then some of them they'll actually squirt back out into the soil. So, anyways, that's what most people are interested in these days because it connects with regenerative agriculture, how plants get nutrients from these soil microbes and the importance of having healthy soils and taking care of your plants and your seed. Microbes are vector on the seed and so forth. Yeah, so Wendy, could you check and see if you got it? Do you have that presentation?
I am. I have it. It's downloading right now. So
it's a big file. It's
big.
It's a big file. It's a big file.
700 some odd. I don't know. We're at 24 megabytes. We're getting there.
It's full of pictures and it has movies in it and everything. It's the I have this idea that this is such an incredible idea that plants are eating microbes that it's so crazy that in order for people to believe it happens, they actually need to see it, right? They have to see the whole process. So the presentations that I give, I always show lots of pictures so that everybody can see what these microbes are doing. And it's so that people can judge for themselves and they don't have to take my word for what's happening. They can draw their own conclusions, about different aspects of the process and decide for themselves. And I kind of think that makes science more accessible also to people because, nobody wants to just take somebody's word for it. We're all intelligent, we're capable of analyzing data. And especially when it's seeable, when we can see it and decide for ourselves, what the conclusion is about that data. So, that's my philosophy about this. So it's still downloading?
It's still downloading.
It's big.
We're Humboldt County has limited internet, so it's takes a little while sometimes. Our download speeds and upload speeds aren't super
fast. That's okay. I thought you California people were really sophisticated. What's going on here?
We're rural up here. We're like farming community. We're super rural. There's no high speed internet. Our high speed internet is like really slow. So yeah.
I actually was there in working in California. I've been there a few trips. I have some company friends that are there, and I've been working with some people. But I did this when I was a graduate student working on fungal endophytes for my PhD. I actually did this great 1 month trip throughout the Western United States. I was at the University of Texas, and I got the University of Texas van for the botany department. And then I took this trip, had 1 month to do this. And by myself, I went through from West Texas and then out through Arizona and to Southern California, and then up through California, first passing into California, went through the Mojave Desert and Needles, California, in that area, and then and then up on the Coast Road 101 before it was too crowded. It's not I think it's a lot more busy now than it was, but it was a beautiful, beautiful road and wasn't too crowded then. And I went up there collecting plants all the way, looking for grasses that had these fungal endophytes in them. So I would go along and I had my microscope there and I would look at these, find the plants, the ones I was looking for, and look at them, make sure they had the endophytes in them, and then collect some and put them in the van. And I was actually robbed in Laguna Mountains. I was climbed into one of the hills into the chaparral country, and I had to crawl down under the bushes to get up. And so I was going up this mountain, and it started to get dark, and it was up there at night. And so I was trying to get down, and I was going back down. I had to climb the chaparral, and then I would fall to the next tree and climb it, and then fall to the next tree and climb it, and fall to the next tree. Finally, until I got to the bottom, and it was just falling right over this cliff, actually. And I didn't fall off. I climbed back on, and I found this way down where there was a sand chute, and I jumped down and got down. But while I was coming down, I heard a motorcycle going away, and I walked down. My clothes were all torn up from the chaparral, and I had scratches all over me. But I made it back to the vehicle, and people had stolen my items that I had there in the van, broke it into it and stole it. So I lost a bit there, but then I kept going. I went on, and my wounds recovered. I finally went into California, Humboldt County. I was into Humboldt County, and remember there this is right on the shore, isn't it? The beach there. There's a beach there, right? I can't remember.
Got a bunch of beaches. There's so up in Arcata and Eureka, there's the Samoa Beach. There's, we're on the coast, so. If you're in Humboldt, it would have been like Shelter Cove area or something along those lines. But up in Northern, you drive right along Crescent City's right on the beach.
I remember that. I camped a night on the beach, and that was so nice. And then I went further up into Washington State, and up on Mount Rainier, I collected some stuff. And then coming down, I actually crashed the van coming down the mountain because I saw a plant on the side of the road, and I started backing up for it. And those ditches are hugely deep. I had no idea. One wheel went off, and I thought, well, just keep going backwards. And then the other wheel went off, and I thought, well, I'll just go over that. It'll be okay. And then the whole van fell on its side and all. And anyways, I had no money then, but we did have this new MasterCard credit card with no expenses on it. So I got a van paid $80 for the van to come tow me out with that credit card, and then I continued on my journey after that.
Yeah, James, the problem was people heard you had grass in your van, and you were in California. That's where you went wrong.
Yes.
All right, it looks like this is downloaded. I'm gonna see if it'll open.
And let's see if we can play it. We'll just go over it real fast so people kind of see what we're talking about.
No, I think I've seen this, and I have a technical question because it looks like you have video of actually the bacteria being ejected from the root tips.
That's right.
How difficult was that to actually pull off? That's a whole other field. Is the microscopy or butcher that word, I'm sure.
The moving parts, making a movie. Well, we have a microscope with a movie camera on it. In fact, we got really good scopes. You could imagine we invest in our microscopes. We live and die by the microscope, right? We live and die by the microscope. So we have good scopes, and we have both scopes actually will have this AVI movie options on it, time lapse and everything. But we just happened to catch it, and that was the lucky. I had an undergraduate student, a young lady who was dedicated to trying to get a movie of that, trying to see it, and so she spent a lot of time doing it. But it turns out I actually caught the video part, but it was just sheer luck to catch it, and
awesome.
And we saw it, and yeah, that's right.
I have that up. Do you want me to hit share screen and bring it up? And then James, you can just say next and I'll click through.
See if you can. Let's see if it works.
All right.
Let's see if it works. And I'll just go over, maybe we'll do 10 minutes worth so it's not too long, but people will kind of see what we're
No,
what
it's
we're
very,
talking about.
and I'll put it in the link to the show too if we don't pull it off, so they'll be able to go back and watch it for sure. Yeah, because it's fascinating to see that video of the tiny. Is that a root hair that's pushing out it, or is that
Those are root hairs. Those are root hairs. So all this action takes place in root hairs. I mean, we think of root hairs as these little organs that just kind of stick out and absorb nutrients. It's not true. A root hair is actually a microbial processing organ, that's processing these microbes and increasing microbes, getting nutrients out of them and extracting nutrients from the microbes and also increasing the microbes and then shooting them out into the soil. I mean, that is the cool thing. So the root hair is a microbe processing organ for plants.
So you mentioned in the talk before how that superoxide molecule strips the cell wall from the microbe.
Yeah.
And then the nutrients are extracted, and if it pushes it back out, is it live again? Are the plants actually farming these things?
Yes, the plants are farming.
Is that amazing? Isn't that amazing?
It really is. Jeff Lowenfels, who's writing a book about bacterial endophytes in plants, he wrote these books of teeming with microbes and teeming with fungi. So he's doing one now about teeming with bacteria. But he says that this rhizophagy cycle is actually a ranching process rather than a farming process, because these microbes, these plants are taking the microbes in and they're taking the cell walls off, like shearing a sheep, right? Taking the wool off the sheep and then putting them back out into the soil to grow some more, to eat and grow some more wool. In the case of the bacteria, they have a lot of nutrients like protein on the cell walls and also minerals that stick on the cell walls. Zinc and other minerals will stick on the cell, even iron stick on the cell walls. And so with protein and these other metals sticking on the microbes, on their cell walls, when the plant takes those cell walls away by oxidizing them, they get all that protein, nitrogen essentially then from oxidizing that protein. And they get also metals, heavy metals and other micronutrients. Potassium actually goes inside, is a lot of the potassium from inside the bacteria as a kind of a solute in the bacterium. Okay, this is it. This is perfect. This is perfect. Winnie, you're a genius.
No, this
You're
is
a
just
genius. we were all trying and we couldn't figure out how to do it. My computer just said, well, my browsing program didn't work, and then I downloaded another one and it didn't work, and then I got a third and it didn't work, and then it froze me up. So let's go ahead. So anyways, this is real fast. I mean, this is basically how plants use soil microbes, how they cultivate soil microbes, how they grow them and internalize them in order to improve their own growth, mostly root growth, but also shoot growth, and how they use those microbes to get nutrients. And I should say, just to make this clear, these plants actually take microbes. They don't leave them in the roots. They take them and they move them all over the plant. So they actually will go into the flowers and the leaves, and everywhere in the plant will have these microbes. So it's not just these microbes. The plant will move these microbes throughout the plant, and they'll put them on seeds so that they're seed transmitted, so that the baby plant, the seedling from the seed, will have microbes on it to help it grow. So let's go to the next slide. So these are endophytes, by the way. An endophyte, just to make it clear, an endophyte, internal phyte plant, is any microbe that goes in a plant and does not cause disease. So these microbes are not disease agents, not like pathogens. They're bacteria, mostly bacteria, what we're talking about, but some of these are fungi. And this actually is a picture, shows a fungus inside a grass. And of course, this is grass, as in the sense of turf grass or forest grass, right? But I don't have pictures here. They show you, but hemp and cannabis grass have these endophytes in them also. They tend to be bacteria. They're all in the seed, and when the seed germinates, they're in the seedling. They're all in there. They're really, and in fact, I will go a step further, and I can't say it's a hypothesis, but we are exploring the possibility that the trichomes of many plants also contain these bacteria, and that those bacteria are part of trichome development. And this is totally unproven at this point, but we have reason to think that many trichomes, for example, in tomato, have bacteria in them, and their development, just like the root hairs, what I'll talk about now, that those trichomes may be triggered to develop by these bacteria. Now, we don't know that to be the case, but it's a possibility. It's something that we're trying to examine. And if that's true, then it's really important for cannabis, because it could affect the THC content in cannabis. And so anyways, that's that crazy possibility that might or might not be true. But you'll see why we think that there could be some connection there. Next slide. Okay, so a lot of these endophytes are bacteria, and they're all over plants, and all plants have them. Next slide. I'll go real fast after these. This is one plant, an example, the giant cactus. This plant, if you look at the seeds, next slide. That's a seed, next slide. Germinate that seed under aseptic conditions without contamination, and you have a seedling like this. And if we looked at those hairs, this is actually stained so we can see those bacteria in the roots, but you can see the root below, and you can see the hairs sticking out there, the bushy thing. Next slide. Those are the root hairs. Look at that. Now that's the root hair, and you can see the little dots in there, the brown dots in those hairs that are crossing the screen. Those are the bacteria. Next slide. So we get a close-up of it. Look at that. Look at all the bacteria in that hair. Those hairs are loaded with these bacteria. All plants do this, especially cannabis. Cannabis roots are loaded naturally with these microbes. So you don't want to sterilize your seeds, because they're in there. They go in the microbes. They go in the seed, and they go on the seed. They're all over that cannabis seed. They're probably very, very important. Next slide. So this whole process was actually not discovered by us. It was discovered by some Australians, some people in Queensland, Australia, at the University of Queensland, and they have a team there that was looking at endophytes. And this young lady here, Channy Pongfu, was one of the investigators. She was actually the author on a couple of these important papers from that group. But they labeled their microbes, some bacteria and yeast, and they fed them to tomato, and they fed them to Arabidopsis. They just put them on the roots, and those plants absorbed those microbes, and then they could photograph them inside the roots. And these are some pictures from their paper. The title of their paper was actually this. It was Turning the Table. Plants consume microbes as a source of nutrients. So their idea was that these plants are eating these microbes. And we've been studying that process since then. And even a little bit before, looking at this oxidative process. Next slide. And so this is basically what we have come up with. These microbes are actually not just going in and being consumed. In fact, some are going in and being fully consumed, but others are going in. They're being hit with superoxide. They lose their cell walls. They're then being replicated inside. They form these protoplasts inside the root cells. They're inside. Oh, but I should say they're attracted to the root by exudates. Those are from the root tip. Plants are secreting sugars and organic acids and sometimes amino acids, and that attracts these bacteria to the roots. Then the plant sucks them into the roots. We don't exactly know how they get them into the root cells, but they suck them in. And then they take their walls off using superoxide. And then The surviving microbes, they shoot out the root hairs. You can see these root hairs with little arrows on them there. That's actually as the microbes will trigger hair growth. And as those hairs grow, the microbes are squirt back out into the soil. And I'll show you all this. And then once they're out into the soil, they'll reform their cell walls. And if they have flagella for swimming, they'll reform those. And then they'll swim out in the soil and get more nutrients. But we know if we take away the bacteria, you get no root hairs forming. So those bacteria inside those root cells are important for forming root hairs. Plants have no root hairs without the bacteria. Next slide. Oh, that we call the rhizophagy cycle, by the way. And these microbes are soil microbes. That's bottom line. But the plant will take them and put them on the seeds, so that they then, and in the seeds, so that they then can vector to the seedlings. Okay, but they're all soil microbes, so that's why you need a healthy soil. So that's why you don't want to, sterilize your soil and then try to grow your plants in it, because you're screwing up really if you're doing that. You want diverse, healthy soils, and you don't want to sterilize your seeds either, because those are the microbes that the plant needs on those seeds. If you do that, you kind of are opening the way up to have poor seedlings, weak seedlings. They get disease all the time, and they just are really not hardy. Next slide. All right, so this is about biostimulant microbes. These microbes are biostimulant microbes. Now, you could buy some biostimulant microbes and use those too. You just got to test them and make sure they're working on your plant. That's the all, because they don't all work the same. Next slide. Grass seed here, and when it germinates, those bacteria colonize the seedling root. You can see they also colonize the shoot, so you get them in the shoot, but they get in the root. Next slide. I'm just going to go real fast over these. Okay, this is a grass root. You can see the blue cloud where those arrows are. Those are the bacteria that this root is cultivating around that. This actually is from a Phragmites, that invasive grass, but all big old Phragmites, invasive reed grass is called, grows everywhere out here. I don't know if you have it on the West Coast, but the East Coast, this grass is invasive and everywhere. But the plant is cultivating these microbes. The plant is, if you put nitrogen fertilizer on inorganic nitrogen fertilizer, you'll suppress this. The plants won't secrete exudates, and they won't take in these microbes. So you have to be careful with putting ammonium, for example, or nitrate, too much nitrate, too much ammonium on soils to grow your plants, because you'll screw this process up. And this process is the way that plants, this rhizophagy cycle, is the way plants are getting their, this is the way they, I mean, you could say, I'm a biologist, definitely an evolutionary biologist, right? So this is the way plants evolve to get nutrients. But if you are a creationist, which is perfectly valid, this is the way God made plants. So to do this and not to get, squirt nitrogen on them, but instead to cultivate their microbes to get their nutrients. Next slide. So this just shows what happens. Okay, if you see that to the left, you see bacteria with their cell walls, and you can see they have rod shapes. To the right, with superoxide, the plant will oxidize off those cell walls, and instead you can see they form these naked spherical structures. Without cell walls, they're spherical, and they're tiny, and the plant can actually replicate these little protoplasts. They actually have a name for them, L forms, but that's a medical name that they invented years ago for these things. But these are what happens inside the root cells. Next slide. These are NOx enzymes that the plants have on root cells to make this superoxide. All eukaryotes have these NOx. They're actually defensive, and it produces superoxide. It takes molecular oxygen, makes superoxide. And this is what they have on the root cell plasma membrane, these NOx enzymes, and that's how they make superoxide. Next slide. Next slide. That shows some of these in roots. Yeah, okay, and this is a diagram of that, and you can actually see the bacteria here. They're actually underneath the cell wall, the gray there, but they're not in the cytoplasm. The white is the cytoplasm there in this diagram, but you can see the bacteria outside the cytoplasm, but underneath the cell wall. They're trapped in there. And the other thing is that the root cells are moving these bacteria around. You can see that little word I have, cyclosis, there, and you can see the arrows. They're shuttling these microbes around and around and around and around. It's really cool what the way they keep those microbes off their toes, off their toes, so that the microbes cannot, because they could get enzymes and get out, but the plant has them trapped in those root cells. And it's super cool process. I'll show you that. Cyclosis. I'll show you that so you see. Next slide. So the microbes stimulate root growth. The plants use these microbes in order to grow their root cells bigger, and also in order to grow root hairs. And I mentioned already, if you remove the microbes, you got no root hairs. And that's the thing I'm going to talk about here, the root hair growth and cell growth. But they also will trigger gravitropic response. And the gravitropic response is this, where roots grow down. If you remove all the microbes, roots don't go down with gravity anymore. They go on the surface or they go up. But then you put the microbes back on and they go down again. So that gravitropic response of roots is really important, that behavior. They don't do that properly without their microbes. Next slide. Tomato is an example. You think of tomato sterile, it's not sterile. It's loaded with microbes. The tomato fruit has microbes in it. In the fruit itself, you've got microbes. And in the seed, and around the seed, inside the seed, there are microbes. And those microbes are, in fact, they came from the soil, or we hypothesize that some of these microbes come from the gut. And a tomato, for example, is adapted to have the seeds go through the gut of animals, birds, for example, or humans, and we think that they may be picking up some microbes as they go through the gut of animals. And the reason we think that, well, first of all, because they're adapted for that. But the other thing is that the microbes we get out of tomato seeds are actually some of the same microbes that we get inside intestines, animal intestines. So, we hypothesize that perhaps some of those microbes are picked up when they go through gut. Next slide. Okay, that's seed of tomato. We germinate that. We germinate those seeds, tomato. We get this next slide. You can get these seedlings come out. And what we've done is we've treated some with antibiotic to kill the bacteria, and some we have not. And you have to be careful. If you overtreat with bacteria, you'll hurt the seedling. But because it'll inhibit chloroplasts and mitochondria. But in this case, we didn't. We treated very minimally. And if you see to the right, you see a seedling that was treated with streptomycin antibiotic to remove the bacteria. You can see you have roots there, and you have no hairs forming. Okay, but if you see to the left, without that treatment, no antibiotic treatment, you can see the hairs forming. Okay, so if you remove the bacteria, we totally inhibit. Okay, and this, you can go forward again. Go to the next slide. That's good. It was good. It was perfect timing. So this shows these, I'll tell you what, you look here to the right at the top of the picture, right corner, and you see no microbes in hair initial, and you see there's nothing staining inside that hair. These hair initials are preformed. And if they don't have, this is on roots, and if they don't have microbes in them, they don't elongate. And so they just stay like that. But if you have the hair initials, and you see to the left here at A, you can see the little brown dots in there. Those are the bacteria inside there. And because those bacteria are there, those hairs, that'll trigger growth. And you can see here, B here, you see a little bit more, and you can see the brown where the bacteria are, right up there. Those are the bacteria right at the tip. And it causes that hair to elongate. And you see it's even longer, and you see there's more bacteria there. All those little brown dots in there those at the tip of the hair. those bacteria are actually replicating when that hair is elongated. Those bacteria trigger that elongation, and those microbes are replicated, and they're also ejected from the hair tip as that happens. And I'll show you all that. Next slide. Next slide. Okay, so why is there this linkage? And this is my student who works on this. Ivy Chang has been working on this, and I've been, of course, we collaborate, all my students collaborate and we do stuff. But you can see here, she's trying to figure out why microbes trigger plant cell growth, why they're triggering hair development. And here, this is a grass to the left that shows this is actually annual bluegrass, a little experimental grass where we removed all the microbes. And you see there's no hairs there on that root. It's stained all brown because we used a stain for that, but there's no hairs on it. Okay, but if you look to the right, we put a bacterium, she put a bacterium, Mr. Royal We, right, Royal We. She put a bacterium on it, and you can see hairs now on that. You can even see little dots at the tips of the hairs. Those actually are bacteria that were squirted out of the tips of the hairs. Next slide. Okay, and we want to know why that is. Our hypothesis is that it's because these microbes are secreting ethylene. And this is a lot of experiments that we've done that suggest that the microbes secrete ethylene, and that's what causes these hairs to elongate. Next slide. It's actually more complicated. And of course, we have to spend our time doing something. So we keep trying to figure this out, what's going on with these interactions. And so this is what we've come up with. It's actually this chemical interactions that happen, and basically ethylene is involved, and then plant with ethylene, then the root cell, the microbes produce ethylene, root cells release nutrients and they grow, but they also produce superoxide, which takes nutrients, strips nutrients out of those microbes. And then the microbes then respond by secreting their own antioxidant, something to counteract superoxide, which is nitric oxide. And that actually ends up being, we think it is a fertilizer then. If these microbes are able to fix atmospheric nitrogen, they can take nitrogen out of the air, and these microbes then have to keep fixing nitrogen and secreting this nitric oxide. Otherwise, that superoxide will degrade them. And these are these chemical interactions. And I'll talk a little bit about those. I'm not going to talk too much about those, but you can see there's some chemistry there, so there's some real, I'm going to do my thing here, real science, right, with the chemistry. Okay, next slide. Okay, so this shows that first interaction. This involves the ethylene, and so you can see how the superoxide reacts with ethylene, and you get hydrogen peroxide and carbon dioxide. That's just a chemical interaction. Those are balanced equations. And we've tested that in multiple ways, and I'm not going to go into all the details, but it's pretty cool what we've done. But I won't go into details of that. You can see that I can answer that if you want. This is a close-up now that shows microbes in a tomato root. You can see the cell around here, and you can see that black arrow, that big black arrow showing that's the microbe there, and you can see the purple around that microbe. That is ethylene. That's stained with ammonium molybdate to show ethylene. Next slide. Next slide. And this is superoxide. You can see it's stained with another kind of stain. We call it NBT, nitro blue tetrazoline, but it stains the superoxide. You can see the purple there. And you see some cells don't seem to have it. This mass out here doesn't have it. Maybe because the nitric oxide is being produced by these microbes and it's counteracting this superoxide. And but we'll go on further. Next slide. This shows the hydrogen peroxide. You can see the microbes. This is an amaranthus plant. You can see all the spherical things. This is a root hair of amaranthus. You can see all these brown. That brown is another stain that shows the hydrogen peroxide. And so the hydrogen peroxide is one of the products in that reaction. And we can see that because of that interaction. Next slide. This is the second interaction. And those microbes they're making arginine internally, and we think that in some cases, the plant just provides them arginine for that. In other cases, they're fixing nitrogen from the air, and they're making their own arginine. We think that the ones that can really survive in this process have to actually make their own arginine, and then they produce superoxide, and that counteracts that. I mean, they're nitric oxide, they make nitric oxide, and that counteracts superoxide. Those combine, neutralizes that superoxide so the microbe doesn't get degraded, and then it produces, eventually it will produce, I'll tell you, it forms this intermediary called peroxynitrite, which is just a brief intermediary, which the CO2 from that other interaction comes into carbon dioxide, and that catalyzes the formation of nitrate. Nitrate is a fertilizer. And so we can give these plants nitrate, but plants normally make their nitrate from these microbes, right? They will make their nitrate, and but if we put nitrate there, we'll make this whole process, we'll make it so they don't need to do this at all. Okay, so they normally will do this. That doesn't provide all their nitrate, but it provides like 30% or more of their nitrate that plants need, and that also has some other effects. Next slide. Okay, this just shows nitrate staining, and you can see the blue here and you can see the microbes there at the tip of a hair. You see the blue there, that's nitrate. That's used as diphenylamine is a nitrate stain that stains blue when there's nitrate there, and so you can see there's nitrate around those microbes. So that's consistent with our model that we showed you, the chemical equation model. Next slide. And this just shows it again. You can see a hair there, and you can see the microbe in the middle, the black hair. The black arrow shows that microbe in the middle. You can see the purple around it. Okay, that's the nitrate again that's being formed in that interaction. This happens to be a cabbage seedling. Next slide. Okay, so with these interactions, actually what's happening is the plant has these microbes in this trap. It's a survival trap for these microbes. And the microbes that survive in this trap, some are going to be fully degraded because they can't survive. They can't produce enough nitric oxide, so they're going to be degraded. But these are the loops. This is the trap, essentially. This is a drawing of a hair. You can see the root hair to the right. You see the little microbes there. There's essentially these loops, carbon loop. There's nutrient or nutritional loops. One is the carbon loop, and that is the microbes are secreting ethylene, and that triggers the release of carbon or exudates from the root cell. And those exudates or sugars go to the microbe, and that's great. But the same ethylene, and this is what happens. Well, the same ethylene will trigger that superoxide. So the superoxide gets secreted, and you can see the next loop. It's called the nitrogen loop. The superoxide goes out to the microbes, and it's going to degrade those microbes unless the microbes can secrete nitric oxide, and that creates the nitrogen loop. This nitric oxide has to be secreted. The ones that can secrete it can counteract that superoxide, and they can survive, and then they get replicated in there. They survive in these cells. They don't get degraded. So the plant has them trapped in here. Okay, if the microbe stops producing ethylene to get out of the loop, then they get no nutrients. They can't get any nutrients, and they're going to die. Or they're not going to replicate. They're not going to grow. They're going to die. If the microbes stop secreting nitric oxide, then they're going to be oxidatively destroyed by superoxide. They're going to be degraded. They're going to die. They have to keep producing this nitric oxide. So that means in order to do that, in order to survive, they must fix nitrogen. They have to fix nitrogen to make the arginine to make nitric oxide in order to counteract that superoxide. So it's a nutritional trap that these microbes are in. The plants have them in its clutches, inside those plant cells, inside those roots. They're trapped. They're trapped. Next slide. There we go. There we go. You see, work or die. There you see, work or die. So next slide. I mean, that's basically what's going on inside these plants. So, I mean, this is just talks about how that could work. It's just, it's a model. It's a hypothesis. These are everything I'm talking about, especially when it concerns nitrogen fixation, is a model for what we think is going on. Everything needs further confirmation. We're confirming it, but we need more science to confirm what we're talking about. Next slide. Okay, this is going to show, this is one of the plants we worked on. This is a sedge from Bonaire, the Dutch Antilles. I love doing this field work because I can do field work in places like Bonaire in the Dutch Antilles. Go there, for 2 weeks and study the plants there. I mean, what a great way to work, right? But anyway, this is one plant. It grows on these rocks. It's a little sedge. It doesn't have soil, but it has a lot of microbes. And I'll show you. Next slide. Okay, this actually shows below you have a picture of the root hair of this sedge, and you can see the little red things. Those are the microbes in this root hair, and this is stained for hydrogen peroxide, which is one of the products in that reaction, that first reaction with ethylene, and it turns brown or reddish. There, you can see the color there. Okay, it's not moving. It's stained. It's fixed. Okay, but the one above is a movie looking at this hair. A different hair, but the same plant, and you can see it's moving. That's the cyclosis. You see the shadows going around. This is how these plants are moving these microbes in these hairs. It's moving them around constantly, moving them around, keeping them off their toes, extracting nutrients from them, and replicating them. Next slide.
Was that real-time speed? Or is that sped up?
That's almost real time. These things are going at about, isn't that incredible? You think of a hair just sits there. It's not true. Hairs are active. If you look at a hair under a microscope, you'll see they move like this. They're moving. Hairs are living things. They're processing these microbes. They are moving the microbes around, and they're breaking down gradients, and they're making sure if a nutrient exchange gradient happens, that slows nutrient transfer. So this, when they break it down, it means nutrients go real fast between them. Yeah, no, hairs are nutrient exchange organs. They're nutrient processing organs, microbe processing organs. What they do is, one thing they do is they'll break these microbes up, and you can see where the arrows are. These are all the microbes in there. These are the protoplasts. And you can see some are big. The big spherical things, the biggest is probably the first microbe in that cluster. The smallest is going to be the last. So these things are replicating from one microbe that goes in. You get many that form, and the older ones will break down. The biggest ones will break down there because of the superoxide is just getting in. They're not able to resist, but the younger ones survive. And the ones that survive are the ones that are able to fix nitrogen and secrete nitric oxide so that will counteract that superoxide. The ones that break down are the ones that can't fix nitrogen and survive. They can't keep doing it. So the plant is selecting and reproducing these microbes that work with it very well. And then it can secrete those microbes out into the soil from the tips of those hairs and repopulate the soil. Next slide. Next slide. Okay, this is a movie. Oops, let's see if it goes. Okay, this is fine. This actually shows what happens is those microbes will go at the, see if you can press play on that. See if you can press play. There's a play. See if you can go, see if there's a bottom, there might be a bar. Okay, that's right. That's play on that one. That's fine. But you can see the little white things at the tip. Those are microbes. They would be moving around. It's those microbes that would secrete at the tip that secrete ethylene and cause that hair to grow. And when it secretes ethylene, there's a growth spurt. And then some of those microbes are shot out and others are pulled back and then replicated again until there's enough to go into the tip. And then when there's enough that it secretes ethylene in mass and then it ejected again. And you have these ejections that keep going. Next slide. Let's see.
Cool.
Okay, so you see an ejection. See that ejection? Okay, it's doing perfect. Don't touch it. Perfect. You can see the ejection now. See that? See the hair? Now, if you look at 3, actually, you can see there's holes there at the tip of that hair. And those are the holes that these microbes are being ejected through. So these hairs, they expand at the base. They start expanding, and then they move those microbes out, almost like a syringe going out. Now that happens to be how these hairs grow. You get this same expansion. So what's happening is as these hairs are growing, these microbes are being pushed out. And then others are pulled back and they're replicated inside those hairs. Next slide.
And again, real quick, is this also real time or is this sped up?
This is real time.
This is real time. That is amazing.
This is real time. Yeah.
Oh my gosh.
This is real time. I should say this is about double real time. So both of those images are about double real time. But that's still pretty fast for both of those, for all three of those images. These microbes go at about 10 micrometers per second. 10 micrometers per second. Think of that, 10 micrometers. I mean, we're talking about a small scale, right? 10 micrometers, but that's a distance that you can see under the microscope. And these microbes every second are passing, this distance, 10 micrometers. So there's a real, I mean, you can actually watch them doing this. Next slide. Okay, so what happens when plants don't have this? Let's see what happens. Okay, this is an experiment. This is with wheat. And what this scientist did, he's a visiting scientist. He took wheat, he removed the microbes, all the microbes from wheat seeds, and he isolated those microbes so he knew what they were and got them in culture. And then he put them on some wheat and some plants he did not put them on. And we'll see what happened to those plants after they grew. Okay, so this actually shows these plants, okay, to the left, without bacteria. This is ones he removed them from and grew the plants. You can see they still grew. It didn't kill the plants, but when he put them on, this bacillus is one of the bacteria. You can see put one of them back on, and you can see, look at the roots, how big they are over here with the bacteria. And the plant benefited. Look how big the leaves are. Plant growth makes a huge difference with these microbes on them. And we know part of that is, okay, this is actually, then he measured nutrients inside the shoots. And here's nitrogen. You can see no bacteria. You can see that's this much nitrogen. You see the bar all the way to the left, no bacteria. And it's less than when he put bacteria on. This is 3 different bacteria that he put on. And you can see it's actually about 30% more with those bacteria, 30% more nitrogen with those bacteria than without. And without bacteria. Next slide. Okay, this is phosphorus. You can see without, all the way to the left, no bacteria. And then you see with the bacteria, you can see it looks like about 20% more when he put the bacteria back on. Next slide. Here's potassium. Look at this. Look, without bacteria, look how little potassium there was. And then he put the first bacterium on, and look, it's like 3 times the potassium. For this microbe, it's really carrying potassium, a lot of potassium, and the plant is benefiting in a huge way with this microbe. Potassium is one of the nutrients that's carried inside these bacterial cytoplasm. It's the solute in these bacteria. So the plant is making these bacteria leak, and it's getting a lot of potassium from these bacteria. It's eating like a potassium pill. And you look over here with the other bacteria, it's not as good. These other 2 are not as good. But still, it's a lot better than with no bacteria. Next slide. So I should say all macro and micronutrients are improved with these microbes, and different microbes carry different nutrients. So it really takes a whole community of microbes, not just one microbe. It takes a whole community of microbes in the soil to give plants all the nutrients that they need. So, and that's the importance of having a diverse soil microbial community. Healthier soil is better. So plants benefit a lot, not only because of growth, because they use it for development and nutrients. I mean, nutrients, like if you look over here, beneficial outcomes, nutrients are one thing, and growth and development is another thing, but plants also benefit because this whole process is oxidative between the microbe and the plant. The plant is oxidizing these microbes, you have all this oxidative reactions happening, and that causes the plants to upregulate their oxidative stress protection to protect themselves against their own oxidants, their own superoxide, right? And the hydrogen peroxide, and everything is being generated, and the ethylene that causes more superoxide. So they are becoming more resistant to oxidative stress. And what that means is that plants become more resistant to abiotic and biotic stresses. Abiotic stresses like salt tolerance and like heat, that all translate to oxidative stress. And so when plants are resistant to these oxidants, oxidative stress, they can grow under more harsh conditions. They become hardier plants. They're more resistant to salt in the soil and heat and cold or whatever else. They're hardier plants. The other thing is that a lot of these microbes will go out into the soil and they'll colonize pathogens, and they'll keep those pathogens from being virulent. They will colonize them and make them leak nutrients, say a fungal disease agent like fusarium. They'll colonize it and it will become avirulent. It won't cause disease. So, and in fact, these microbes will take nutrients from these other pathogenic fungi in the soil, and then they'll carry them back to the plant, make them available to the plant. So this process benefits the plant. So one other benefit that's very important for cannabis growers is that these microbes also will impact the nutrient quality of plants. They will make those plants have more, for example, carotenoids and carotenes than without these microbes. So squirting nitrogen, you'll have less of these quality nutrients than when you have these microbes present. And I would add, because it has not been tested. Actually, I've heard there is a paper. I haven't seen the paper that shows that the microbial community does affect, and soil health does affect THC content in cannabis. And I have not seen the paper. I just heard about it a couple days ago. I haven't had a chance to actually look for that paper yet. But there you go. Next slide. And if you're interested, these are some papers. We try to publish these papers that are of general interest in agriculture to the industry, horticultural industry, and other companies, and for general public. We try to publish these in open access journals. And it costs us more, but we do it because we think it's important. But you can get some of these articles on the internet from these open access journals. And one of them, if you just Google rhizophagy cycle and the journal Microorganisms, you'll come up with this paper. And it's open access free. You can get it. If you Google, say, for example, my name, White, and then endophytic microbes, and maybe pest management science, which is the journal Pest Management Science, you can get this other review on endophytes, and it'll talk about more of this. And next slide. And I just have to say, if it's not obvious already, this is not a one-person show. There's lots of students who were involved in this work, different aspects of the work. There's visiting scientists, there's postdocs. There's a lot of people over a long period of time that have been contributing to this work from multiple countries. And so they all, some of those are shown here, some of those are listed here. Some may not be, but they've all contributed to this work. And there's people, I should say, on an international basis that are working on bacterial endophytes and fungal endophytes. There's a whole endophyte community that I should say over the last 40 years have contributed to this knowledge. And, this is just a piece of it, an ultimate, piece of that work, but there's a lot of other investigators involved. So that's it. That's all I really have for the presentation. So any questions?
I got a couple things I would like to say.
Yeah, whatever you like.
That is awesome. And it is so important, I can't be emphasized enough how important that work is. I mean, agriculture is the foundation of humanity, and I'm hoping big agricultural schools are looking into this. Is this material, like in Nebraska or are they looking at this other than just let's feed things potassium nitrate or whatever?
So there is more and more interest in regenerative approaches to agriculture. So I can tell that, but I have to also say that this work comes to you from a field that most people are not tuned into, and that is plant endophytes. And most people are not studying endophytes. You get people in every country that are studying endophytes, and they're tuned in to what I'm talking about. But other people are coming. This is really important in regenerative agriculture community and organic agriculture community, because it shows they've always felt that soil microbes were important. And this is one way that shows why those microbes are important in the soil food web, a piece of the soil food web that is important for plants. But it's bigger than that. And, will many of those, how long will it take before this is mainstream in agronomy? Before all agronomists recognize that it's something important to consider? I don't know. Some of that things filter into knowledge in sometimes in a different ways, and sometimes you have to have. And in this case, the general public has embraced these understandings of science. And industry, and I would say also agricultural industry, has embraced the science. It's a little bit slower in coming in terms of, say, agronomists, but it takes time to change. They've been in, chemical kind of approaches to agriculture for a long time, and it's kind of hard for them to change the way they think and the way they do research and the way they operate. And you can understand that, conservative. It's conservative. Science is also very conservative. And agronomy, I would say, is one of the most conservative fields that we have in science.
There's a pun there. So,
you went right over my head. You went right over my head. You better, let's see, most conservative, the pun.
In the field.
Yes, that's the pun. See, you were a teacher. That's obvious, so you're very sharp mind to pick out these puns here.
Yeah,
I'm a teacher too, but I'm not as quick as you are. It takes me. I have to study it a bit. I've got to study it a bit before I get it.
And before Wendy, thank you so much. You saved the day being able to show that slideshow. That was beautiful. I'm so glad people got to see that. I'm going to put a link to it into the bottom of the show. But Wendy, I know there's some good questions coming out in the chat, and I know Wendy might have some good questions. So I'm going to try to button up a little bit. And James, that was beautiful. That was awesome. And I think your work cannot be. It's understated to say that it's important. I mean, and maybe that's something the cannabis community seems to be much more in tune with, is getting into. Oh, look at that. Is getting into these regenerative and healthy living soils. So maybe we can be a kind of a forefront to that. But anyways, yeah. Wendy's actually big in that. She runs a really good group on Facebook, Organic Cultivators, that this is their main focus. So I think this is a great symbiotic relationship.
I love that. And I love that you're focusing so much on bacteria, because a lot of what we're kind of looking at, especially in the cannabis industry, is a lot of these endophytic microbes that are fungal, not necessarily bacterial. So this is a really different kind of spin on things. And my brain is immediately going to nitrogen fixing bacteria. And, so just a little bit of my background is I practice Korean natural farming. So we do indigenous microorganism collections out in the field and propagate those out to make larger and larger piles to bring into our field to create diversity. Mostly that is focused on fungal diversity, but there's also, of course, terrestrial yeasts and bacteria that are inoculated as well. And what I found on my farm is that the more I do this, so we're about 3 years in of doing this pretty sincerely, and more than that on my home garden as well, which I don't even fertilize anymore. I literally will toss a few, like, handfuls of my rabbit manure, and that's about all that my plants get at home. And I see zero deficiencies pretty much across the board. And so for me, just experientially, I figure that, all that diversity that I'm including has things like nitrogen fixing bacteria and, all of those great microbes that are out there in nature I'm bringing in, and then they're finding happy spots. And, I continue to inoculate with that, but I don't really feed a lot. And so I know with cannabis, a lot of people are growing in potting soils. They're growing on cement in, smart pots and things like this. And it really limits the diversity that's in your soil. And even when you're inoculating, a lot of people aren't doing these indigenous collections. They're going out and buying products off the shelf. They're buying Mycos and Azos, which are extremely limited, or they're buying other inoculants, which have, maybe 5 or 6 bacillus species, and then maybe some endophytic microbial fungi and propagules of this or that or whatever. But it's still fairly limited, it seems to me. Like maybe there's 12 to 20 different species of fungi or bacteria in that particular product. And then, of course, there's the question of how much of that is actually viable, et cetera, et cetera, et cetera. So I just, for me, I'm thinking about, for the people that don't want to do the collections on their own, are you guys playing with, some of these products that are on the shelf? Do you know anybody that's doing testing on that type of work? Are there different species that you guys have found that you specifically think that we should be looking for? Just kind of a broad scope of what that looks like for somebody that doesn't want to, collect things from nature.
Yeah, we are looking at individual microbes, and some of those have been picked up. Getting them off wild plants, for example. Some of those have been picked up by companies like Indigo Ag, and that's nothing. I'm not telling any secrets here. Indigo Ag gets a lot of their microbes from academics. That's kind of their model for obtaining microbes. But we also have been working with some companies to test out products, and we have some. I can't mention anything, obviously, but we have information about how those, and most of those microbes that companies produce, or many of these microbes that companies produce, are in fact endophytic, especially the bacillus species that they produce. Now, they're not all equal, and you could gather that from the work that I showed on wheat, where some microbes work better than other microbes. And that's exactly what you see. Some microbes are really good and some microbes are not. And you have to be careful because some microbes can even be inhibitory. So I won't go into any of that. I will say that in terms of, I will share one little thing here. The indigenous Americans, the Indians, used something called the Iroquois corn medicine. The Iroquois used, they grew corn, and they realized that their corn could use some, they called it medicine or magic, right? And basically it was microbes. And what they did is they'd go out and get wild grasses, and they would take those wild grasses, the roots, and they would boil them in water or put them in warm water. They didn't necessarily boil them, but they'd heat the water, and that would break some endospores, because that breaks bacillus endospore dormancy. That would break those endospores and get the endospores into the water. The bacteria into the water, and then they, from those roots, those wild grass roots, and they put their corn in it, partially germinate their corn, and then they take that partially germinated corn that had then acquired those microbes from those wild grasses, and they plant that in their mounds, their corn mounds, where they were growing their three sisters, right? And then they would produce that. They would do the Iroquois corn medicine. Of course, that gives them a whole community of microbes off those wild plants. It's not like our 1 or 2 or 3 or 4 or 5 microbes. They would get a community, which is more like what you're doing when you're using the Korean agriculture approach. So, so they all work. But here's the other thing: you need to always experiment. Don't trust that a product is going to work. You try it, make sure it works. There was another question that I could address real quick. about using sterile hydroponic approaches. Anything, what that would do actually is that prevents the plant from fishing out any new microbes from soils, from diverse community and soils. You'll still likely get the microbes that are seed vectored, so your plant should be okay, but you're not going to get the benefit of collecting new microbes. And if you have nitrogen in there, you may also reduce the internalization of those microbes that were seed vectored. So you may have seeds that have fewer and fewer microbes vectoring on them. I would think you would want to test that. I can say that now, but I would think you would want to test that and see if after a few generations of seed production only in sterile hydroponic, if you see some reduction in seedling hardiness, or if you could look at the actual microbes in those seeds, if you could see any reductions in the microbial diversity or the microbe numbers that go into the seed, or you'll see a reduced performance in your seedling if you do see reductions in the microbial community in the seed. They'll become weaker in time, but you have to test that to see for sure. Theoretically, though, you should see loss over time.
And for people who don't have microscopes, so I have a soil microscope. I'm kind of a, amateur soil scoper. I just love staring at the stuff, and I don't know as much as I would like to, and eventually I'll get there, I'm sure. But there's a lot of people that don't own their own soil microscope, or they don't know how to use them. So would your recommendation for those people be, so I'm a huge proponent of some real standard scientific trials, like limiting your variables. So we see a lot of, especially with cannabis growers, people growing from seed, where you have such a wide genetic range between seedlings, it's really hard to do some of these trials. So when I run my tests on my farm, I always make sure I'm doing clones so I can limit my genetic potential, my genetic variables. And then I always have a control group, which I don't do whatever it is on. And then I'll have, like if I was going to test, kind of like what your bar graphs were showing, like here's the control that didn't have anything, and here's this bacillus species, and here's another strain, and here's another strain. So now we can see exactly what species is doing what. And I know that for some people, again, that don't have a microscope or don't do soil testing, there's a lot of places in the United States and in the world, indeed, that are still very, very illegal. So it's not like people can really send things out. They're doing this secretly in their closets. Would you just recommend being able to try to track that visually? Do you think that these microbes are going to make enough difference that visually we'll be able to see it that distinctly?
Yeah, I think that's the key thing. You look for seedling performance. That's the key. That's the really the important outcome, right? Important outcome is how those seedlings perform. See if your roots have hairs. Are the hairs bigger, with a certain treatment than with the other treatment? Do you tend to have? You actually can see. Does when you put the treatment on, do you get less root development? That's an indication that you've got some, and you may need to grow it for a while in potting mix to see if that's the case. Wash those roots and look and see, are they bigger, or are they equal, or are they smaller? Can you see root hairs when you have your microbes there? There is another way that I could say people could do this, and it's kind of interesting. We've been trying to figure out what the chemical interactions is between the microbes and the plants. But one chemical that we used is something called silver sulfate. And silver sulfate, when there's more bacteria in roots, silver sulfate will turn blacker. Like say, for example, you made a silver sulfate solution. Roots get darker when there's more microbes in the roots. So you can actually stain them with the silver sulfate, say 1% silver sulfate, 1 gram per 100 mils of water, or something like that. And you could see, just germinate your seedlings and then stain them compared to roots you have not stained, or even you could even do sterilization to see if you could sterilize the microbes and see what happens then, microbes on the seeds. But then you could germinate those on, say, paper, for example, and then stain that, soak it just for a few minutes, 5 minutes in this 1% silver sulfate. And the blacker means there's more microbes in there inside the root. And really, when the microbes start to reform their cell walls, they'll turn black there. So that's where the dark comes in. And so there's more rhizophagy cycle. You'll have more microbes, more black. The roots will be blacker, darker. It's really, really interesting. So it's a quick spot test you could do. You could play around with that and see if you could get that to work, and see if you can differentiate, with and without good soil. But that's a way. You don't need a microscope then. Just look for darkening of the roots.
That's awesome. I know Patrick just put up a question that was somebody's wanted to know your opinion on EM1. There it is, Terraganix EM1 as a seed inoculant.
I'm not an expert on everything about, biologicals you can put on plants. So tell me what this Terraganix is.
I honestly don't know. I know EM1 is an inoculant
EM1.
that people use a lot, right? And it's I don't really know that much about it. I don't use it, so. Patrick,
I don't know that particular what it is. Maybe they'll follow up with it. I have a couple questions. Recently, I did some experimenting with old seed germination and also tissue culture. Now, I've heard you talk a little bit about that, and one of the things, a lot of the, they say sterilize the seeds with hydrogen peroxides, some kind of 10-to-1 hydrogen peroxide, or there's a lot of different methods. And actually, I found none of them really worked on old seeds. I kind of failed on that experiment. But from your point of view, if you are doing tissue culture, which I think is huge in especially ornamental agriculture, would it be best to reintroduce some of the biology back into that, or like I'm guessing you'd suggest not to sterilize seeds, or
I would suggest not to sterilize seeds. Oftentimes, the old seeds, the reason they're not germinating is that they've lost some microbes. And this is actually comes not from me, but from a biologist at Salisbury University in Maryland, by a scientist named Professor Mark Holland. And Mark spent much of his time studying an endophyte Methylobacterium species in plants. And what he's found is that a lot of grasses, particularly a lot of plants, when they get old, they lose their germination capability. And what he could do is he takes this Methylobacterium and he can put it back on the seeds and then recover that germination capability. So what appears to happen is these endophytes appear to be dying in these, because Methylobacterium actually will go in the embryo inside the seed itself. And it will die over time because in the seed there's not a lot of nutrients being released, so they just run out of food and die if they don't germinate, the next year, the next season. And then they don't germinate properly, they don't function properly. So what he's shown, in fact, he patented that process for taking that microbe, Methylobacterium, putting it back on seedlings and then recovering the germination capacity. That's been a patent out that's been a while now already. But Mark Holland is another one of those endophyte investigators that I'm talking about. That he does know about about all the stuff we're talking about, and has his own experimental line.
So really quickly, getting back to the Terraganix question, somebody said it's 3 things. It's not. It's their label says it is 1 million CFU per milliliter of Lactobacillus casei, CASEIKC. I'm terrible at pronouncing things when I read them. And then basically it's 99.99% water and molasses. So in my opinion, this is probably not something I would spend money on, because I'm not going to buy a big bottle of water, especially sugared water, which is really all that is, and some lactobacillus. But yeah, that would be the specific strain of Lactobacillus casei.
So let's see. There's 2 things here, 2 points I want to make. Okay, you have to try the product. I see that. Thank you, Wendy. There's two things that I want to mention here. One is you do need to try the microbe, this lactobacillus, and see if it is participating or if it's improving your plant growth. But there's another way that that product might improve plant growth, and that is that sometimes if you don't put too much on, you don't put too much carbohydrate on, too much molasses, the molasses will go into the soil and will stimulate microbial growth. And those microbes that you've stimulated from the natural soil will release more nutrients, for one thing, from that soil, and the plants are still absorbing nutrients from the soil, but those microbes that are increased also can then, some of them at least, will participate in the rhizophagy cycle, and so the plants might be able to get those. But that's an effect, a carbohydrate effect that's independent of the lactobacillus. And so you have some people in regenerative agriculture that are using sugars or molasses on soils but you have to be careful, because if you put too much on, you're going to inhibit your soils. You're going to inhibit your plants. Too much sugar will actually cause some significant problems for your plants. So you want to be very careful with how much you put on.
You're on, Patrick.
There we go. I'm trying to do too many things. So you mentioned before there the root will put out a little product to attract bacteria or the microbes to the root. So so I've heard this thought before. If you're adding sugar to the soil, it's not it's taken away from the attraction to the roots. Is that something to think about?
No, why didn't I say that? Yes, the big problem is that with all the sugars in the soil, if there's too much sugar, the microbes cannot find the plant. They can't find the plant, so you don't get internalization of microbes there. They're not attracted. That actually is interfering then with the rhizophagy process, putting too much sugar in the soil. So you want some sugar that's not going to be constantly available, that'll break down. Microbes will break it down and then it's gone, so that the plants can then attract these microbes to the roots using its own sugar. Yeah, interference, it's an interference phenomenon where you interfere with the exudation, the role of the exudates to attract these microbes to the roots.
Okay, I have a question if you don't mind me asking, because like I said, when I started looking at the research that Rutgers was doing, I was looking to how does a plant root absorb a phosphate molecule or a phosphorus molecule, and correct me if I'm wrong, but plants take what they need when they need it. So what I've heard is like there's a little protein on the root hair or the root that rotates out, and it'll grab, let's say, a phosphorus or a nitrogen or a potassium, then it rotates back in and gives it to the plant. So what really blows my mind is I was thinking that a root could maybe, I don't know, absorb things with 10 molecules, 10 atoms, 15 atoms, but if it can absorb a bacteria, which has billions of atoms in a bacteria, that's fast. So how big are the holes in the root that can do that? Is it a protein that pulls them in, or what's going on there? Do they know that?
The short answer is no. We have some theories about it, and I'll tell you there's three theories actually that are out there. And one is, and this is from the Australian group, that the microbes are secreting, and this is other evidence to think that this is the case, that the microbes are secreting their own enzymes to enter those root cells, and once they get in, of course, they become trapped inside the root cell, because the plant then has them in its clutches, right? And it can't get out then, it's exposed to superoxide. But that's one theory, and there's a lot of evidence to suggest that. And another theory is that the plant is manipulating these microbes using chemistry, using organic acids, and causing these microbes to infect the root cell I mean, the same thing happens in human gut when you have like IBD. You have collapse of organic acids, and then you have the microbes that will then become very aggressive and penetrate into the gut tissues. Okay, so that something like that may be happening on root tips, where the root tips themselves are absorbing these organic acids like butyrate and propionate, and then that's causing the microbes that it's cultivating there to become more aggressive and then to penetrate into the root cells. That's the second theory. The third theory is that these microbes are simply pressed into the cell walls of the root cells. And the reason for that is that those cell walls are thin. And we've done some experiments. We had a visiting student here from Pakistan, actually, a nice young lady, Sadia Bashir, and had a lady actually from Brazil also that did some work with nanoparticles. And with nanoparticles mixed with the microbes, it just pressed the nanoparticles and the microbes. I mean, the nanoparticles went into the cell. And of course, unless they were sticking with the microbes, they're just pressed in because they have no enzymes. So it's possible they just stuck on the microbes and they were internalized into the root cell. But it's also possible that the mechanism of entry is just physical pressure or some electrostatic interaction that happens between the microbes and the root. We don't understand a lot of what's going on down at the micro scale. So, yeah.
From a physics point of view, nothing moves unless it's pushed or pulled. And it's either gravity pulling it down or a plus or a minus charge, right? That pulls it into the root.
That's
right. The only
well, it would be, there is some thought that it could be electrostatic interactions that are happening down there, some charges that are pulling them in. We don't know, right? We don't know. We do know that plants have these because you can measure them. You could take a plant root, for example. We did some experiments with plant roots, with the mangrove roots. You can put a voltmeter on either side. I mean, from the center of the root and then from the edge of the root, you could actually measure voltage. And so there's a current that goes from those roots outside to in or inside to out, or something that could make charges that could be responsible for pulling cells into or particles into root cells. We don't know. All that has to be explored. Nobody does that kind of work. I mean, nobody does that kind of work.
You're on mute again, Patrick.
I know, I'm bad. Do you have a link for can you read that up there about the Native American methods?
Yeah, I could tell you there's a book by, this is Parker, I think it's called Parker on the Iroquois, which was Parker was one of the Indians, I think, that became he was a writer, turn of the century. He became an Indian, U.S. government, Indian government Secretary of Indian Affairs or something like that. And he wrote some books. And one of them describes that process, Lonesome Lake or something, I forgot.
So Arthur C. Parker, I believe, is the author you're talking about.
Yeah,
he has quite a few books. It looks like there's definitely Iroquois, the Iroquois one that you mentioned. He's got a lot of really cool books. Iroquois uses of maize and other food plants, perhaps.
Look at some of those. You can buy those on the on the internet still. They make some of those. Parker on the Iroquois, something like that.
Yeah, Parker on the Iroquois is one.
Iroquois. See if you can get some of those books, or you can find them online. You can just, I think there's Google versions of them. See if you can find that description in one of those books. It's really interesting. And of course, they didn't know that it had anything to do with microbes. Iroquois corn medicine was magic to the Iroquois.
Remember how you do this?
Yeah, it's available on Amazon. Parker on the Iroquois, New York State Studies by Syracuse University. $17 for a paperback on Amazon. So for people that are looking into it, that could be something cool. Yeah,
Thank
it
you,
looks like
Wendy,
this
for
is
doing that.
basically sorry, what was that, Patrick? Am I
Oh,
lagging?
thank you. No, thank you so much for following up on it. The internet is so powerful that you could just fact check
can.
me. That's pretty. That's
Fact check it. Yeah. It came from, it didn't come, I didn't find that myself. That was, I've seen the literature. I got the book myself and I looked at it in the book, but it came from Walter Goldstein. And Walter Goldstein is a Wisconsin corn breeder, breeding corn that is has microbes in it, organic corn, right? He's corn for organic corn production for producers, and one of his goals is to get microbes, nitrogen fixing microbes, back into corn. And so someone pointed it out to him, and he shared it with me about that, about the Iroquois corn medicine. So it's definitely out there. What you'll discover, and this is just an insight, is that previous cultures and previous peoples who thought they were doing magic did things that had to do with microbes or had to do with chemistry that we know is like the people doing witchcraft would use. They would use datura and other plants. They'd get the alkaloids out. They were doing chemistry to do that, to go visit, do their, have their visions and the psychedelic journeys and things that they had. They didn't think it was chemistry at all. They thought it was magic. And so no one bothered writing it down that there was chemistry there. It was all magic because they didn't know it. It's magic. And of course, if you're writing history, this is evil stuff. And so Indian magic is as bad as, witchcraft magic or whatever it was, So they didn't write that. So all these things, people were doing this really sophisticated stuff. They didn't know it. It went into another way of thinking about things, a magical kind of interpretation of the universe. But it's doing what we would do when we get our microbes and we put them on. It's using these microbes, or it's using the chemicals that plants are producing. And so you don't always see that written in the literature. You'll see they're doing something. They might mention it. They might not. They might not mention they have that plant at all. Instead, it's something magical that they did before they ever had the magic, before they ever did their experience, whatever it was. Yeah. You're right. Also, full circle. You're absolutely right. Full circle. Absolutely right.
I agree with that. Right. Yeah. Before we get too carried away, you've been generous, and I think we're all working, and we've been an hour and a half, and I know there's been some good questions. So you let me know when you want to wrap this up, because we could talk for all day. These are fascinating subjects for sure.
Well, we can do some more questions, and then I probably will have to get to work after we do. I don't know.
Okay.
4 or 5 more questions.
Okay, cool. If anybody has any questions.
I was going to say, there's been a lot of side chatter about cracking old seeds, and especially for cannabis, a lot of us have old genetics that have been stored. And I've done quite a few side-by-sides, and the best thing I've actually found is a seed treatment solution that's Korean natural farming. And it kind of makes perfect sense because you're using a fermented plant juice, which is high in hormones and microbes. You're using OHN, which is kind of a homeopathic immune booster, if you will, is kind of what we liken it to. And then vinegar as a catalyst and pH balancer. And even then, I've had some old genetics that still were really hard to germinate. The seed shell, the outer coating just gets so hard, and the plant the seedling inside loses its vigor, so it can't really crack through. So in that case, for those of you out there looking to try and do this, there's a few techniques that have helped, but it's still kind of hit or miss. But using a matchbook that you put sandpaper in, so you can scarify the seed to kind of weaken that husk, that shell a little bit. Or I guess they also make seed crackers. I've never used one, but it's basically a little like clamp that you squeeze down and it just kind of cracks the edge of the shell. I've had people say they've gotten decent success with that. The problem with some of those old genetics is the seedling itself tends to really lack vigor. So I've had some that are about a decade old that I've had amazing success rate using the seed soak solution and a little scarification. And I've had some that are a bit older that I got to germinate, but they still were just they did not ever, the seedlings were crap, and excuse my words, but they really were. And then the plants themselves had a lot of genetic things that were not, present originally. So I had larger rates of hermaphrodism, things like that. So just for people out there that are trying to crack old seeds, there are also companies that are doing like tissue culture and things, which people say can be a better track. So if you have the money and can throw something like that towards somebody that's doing that on a much more professional level, I would probably recommend it. But I don't know. And then
I see the question.
Yeah.
Question. So what are the best bacteria to use for rhizophagy cycle? The bacteria are going to the plants are going to select the microbes that they want. And they'll pull out of the soil the bacteria. Now, if you're going to, bacteria like Bacillus works, but again, every strain is different. So you have to make sure it works with your plant. And I can't say. I will add that, you have to experiment. And nobody can take that away. No one can tell you, oh, this is definitely the thing to do. You're going to have to test it. Don't trust anybody. Test it. Test everything. I will mention one thing about the seeds dying. Microbes dying in seeds. With cannabis and many dicots, you have those microbes vectored right around the embryo, right around that nucellus tissue. The biology of the seed development, you have a little ovule, and then you have this food material inside that ovule, and that's the nucellus. And then you have the embryo sac and the embryo that develops in that. So the microbe is in that nucellus tissue right around the embryo sac. And so if you take that away, if they die, say after 10 years, if the microbes there die, then you're going to have weak seedlings. And don't forget, the microbes play a role in cell growth. So those cells won't grow, they won't expand. And if they expand, they're not going to, even if they break, they're not going to have the microbes in them to make hairs and so forth, so they won't grow right. And I saw a question pop up now. What was that question again? Did somebody catch that? Somebody catch that last question. Can bacteria, fungus replace pesticides? I wonder. Our endophyte wet dream is that these microbes can be used not only to cultivate plants, but also to control fungal diseases, because that's what they will do. They will go out and colonize fungi. Sometimes they'll just go on the surface. Other times they'll actually go inside the fungal hyphae, and they'll make them avirulent. So they will control diseases. Some of those same fungi then can grow onto the plant as an endophyte, even though they're disease agents. But with those microbes on them, they don't cause disease. Instead, they'll have a beneficial effect or no negative effect, certainly no negative effect on the plant. So having these microbes turns everything around. And in nature, there are no fungicides. Those plants that grow out there in the jungle, out in the woods without disease, they're growing there because they're hardy, but they're also growing because they have bacteria and basically beneficial bacteria as natural part of their structure, and all around in the soil and everywhere in their cells, around their meristems, around their roots, in their cells. They have these microbes. So yeah, you can use, in theory, you can use microbes for plant cultivation without using anything, no fertilizers, no fungicides, even no herbicides. The right microbe will inhibit competitor weeds and not inhibit your crop if you get one that's adapted for, say, cannabis. So in theory, you don't need any of those chemicals at all. But of course, that's a theory. That's our, like I said, an endophyte biologist's wet dream. So, anyways.
No, actually, I got a lot of my philosophy from I don't know if you've been around Crescent City. We have the most beautiful forest, I think, in the world, the redwood forest, and nobody fertilizes them and nobody puts pesticides on them, and they're absolutely pristine and healthy and beautiful. So, yeah, nature's figured this out long before we have, and I think that full circle is absolutely true. And just one point I like, Wendy touched upon it, and you did too, is this the scientific background helps in the sense that you have to be disciplined to change only one variable. You know, the people who try 3 or 4 different things, it's almost meaningless. So it takes a lot of time, but it's the quickest route. There's a pun there too. The quickest
root. Okay, I got that one. Root. I got that one. Okay.
The quickest route to
Okay.
to progress is being disciplined and changing one variable at a time, and it's not easy to do for any means, but do that the best you can. There's one more question up there. You've been so generous. I would love for you to come back on again sometime. But anyways.
So I see this question. Patrick, I see it.
Okay.
Do you have weaker microbes grown in the lab? I mean, everything is theoretically possible, right? I mean, they could lose plasmids or something like that. But the tests that we have done with biostimulant microbes that companies have is that they do become endophytes, and that some of those biostimulant microbes do have beneficial effects on plants in terms of promoting plant growth and making roots that grow significantly and carrying nutrients into plants, where we've measured those nutrients into plants. So even if there was a weakening from using a laboratory microbe versus a soil microbe, you still have those benefits, and we can't, I mean, our evidence suggests that. Now, we never really looked at the question of, do they lose, some strength by being grown in the lab? In theory, you do get plants like fungi, for example, will get attenuated in culture as you keep growing it in culture, and then eventually they won't sporulate, or they won't do what they need to do. And that may be where they're losing some things. They may be losing because fungi are complex, and they may have multiple nuclei in there, and multiple copies, especially Ascomycetes, multiple copies in there, or Psidiomycetes too. They'll have multiple genes in there, but they also will get bacteria that go inside and plasmids that go inside. So you could have those kind of losses. But I can't say that the biostimulant microbes are not still good, because they are from the ones that we've tested.
So I believe what that question is pertaining to is that there's been some chatter in the community about purchased microbial inoculants that have to be continually applied because they don't actually continue to self-propagate in the soil or with the plant. And so you can add a very large amount of certain bacillus species or certain endophytic fungi, and they'll only be there, like the population, if you add 1,000 CFU, and then you'll test in, or, I don't know necessarily where this comes from. I know some of the people who it comes from, but I don't know where they're necessarily getting their information. But what they're stating is that within 6 months, if I was to add, a microbial inoculant, those species that I've added are no longer present in the soil. And so a lot of these things you have to continually add throughout the entire growth cycle, because they're not actually stabilizing in your soil or your growing medium. Again, I don't know for certain if this has been proven out, or who's doing this work, or if anybody is doing this work. But I believe that's kind of what that question is pertaining to.
So I could say something about that. There is no substitute for having a healthy soil. There's no substitute for that. And for having healthy seeds too. There's no substitute. That's where your microbes, your diverse community comes from and keeps coming from. That's the original source. So all these biostimulant microbes, all they do is they will colonize the seedling, and they'll get things started. It's like a kickstart, right? It's kickstart. You got to make sure it works for a kickstart, but it's like a kickstart. And they may or may not outcompete the soil microbes. Once you get a root system going and a lot of microbes stimulated out in the soil, that's different. Then you can take over, then these other microbes take over, and the plant's getting the ones it needs, and they may be better than the ones you put on. You might want to get rid of those first ones because they might get rid of the better ones that come later. The problem we have is that a lot of our seed production and seed collecting and storage technologies are not conducive of developing a healthy seed microbiome. So you're at a disadvantage when you use our modern seeds. Seeds produced in nature are totally different. They have typically you have a healthy microbiome that develops. They stay in nature, they get rained on, they fall on the soil, they get more microbes on them. It takes time, and they develop their own microbiome. But when we take seeds and we put them in a dry place, we limit that microbiome development. Put it in a cold, dry place, we limit the microbes that even grow inside the nucellus, inside the seed, right? So we may need something to kickstart that rhizophagy cycle. Then once that happens, they start getting a lot. And the other issue is that people, if they're looking at soil microbes and they can't find it in the soil, some of those microbes will go into the root cells themselves around the meristem, and then they'll go in those meristematic cells, and they'll actually continue to develop and replicate in those meristematic cells, so that then they will actually be inside the root. Now, I can't say for any particular microbe, but that's what some of the microbes do. Now, the biostimulant microbes, particular ones, I can't say. They could, however, be inside root cells and not going into the soil per se. So that can also happen.
So you're saying I get to get another microscope?
You can, Wendy, but I got to tell you, it's really it takes stains, and it takes really good microscopes. I told you we live and die by the microscope. Our cheapest microscope is a $15,000 Nikon. Our most expensive microscope is not the most expensive one you can get, but it's a $25,000 Zeiss. So we really now you guys may be able to afford it. You got the cash crop, but that's a lot of money to put into a microscope. You got to be really.
I don't need a new car. I'd rather have a new microscope.
You don't need a car, yeah. Well, it may be up to now $40,000 for this Zeiss. So you may be talking about a really good car. But
there's a possibility that the Safe Banking Act has passed, so maybe I'll be able to get a loan.
Yes. Yeah, and there's a time-honored, secret horticultural production. So there, you don't have to do that anymore. But even in New Jersey now, we have legal marijuana. I guess. Maybe cultivation, I don't know, but possession is legal, so they're just figuring out the laws right now. Yeah.
No, there's a lot of momentum in our favor right now.
Yeah.
Yes. That banking bill, believe it or not, is a big deal. It's a huge deal as far as it's huge. Little steps, but we're getting there. And I'm so glad you're talking about cannabis, because it is a one thing about it. So it is a powerful. It's money wise. It's worth a lot of money compared to a lot of crops.
So the big question, of course, is can you get different microbes and enhance the chemical composition of cannabis, and particularly if these microbes are stimulating hair development, like they do roots. Like they do the root hairs, if they are stimulating that hair development, they may also be affecting the, and that's an if, you could say, okay, here's another hypothesis. It's another hypothesis, but that's what it is, a hypothesis. If it is there affecting that, then it could be very important.
Well, it's fascinating too to see that picture of those root hairs and the bacteria with that ball on the end of it that's so reminiscent of the way that trichomes are built on the plant. So that's for me, that's incredibly fascinating and really brings into, for the cannabis industry specifically, a lot of questions about what we are allowed to do and not do with testing. So some places like Colorado still have a total yeast and mold test that they can fail very, very easily because it's a very low threshold. So if you're doing organic cultivation and you're using something like an actively aerated compost tea or a bacterium or something and you're spraying your plants, you can fail testing even though, maybe there's really nothing actually pathogenic or, human dangerous for humans to consume that's on those leaves. So the more that science can start showing this type of stuff, if that is true, and I get that it's totally just a hypothesis, but if we can start seeing things like that, it really brings science into policy and regulation and allowing us as organic cultivators to go further forward with the processes that we'd like to be able to do that we're somewhat restricted from. So I for me, that was like really cool to see that visually. I super appreciate your showing us that and sharing that presentation. That was great.
That was awesome.
And like I say, in tomato, which have these glandular trichomes, as those tomato seedlings develop, we see microbes in those trichomes and actually emerging from those trichomes. And we have with a colleague of mine, a young colleague of mine in Colombia, down in Cali at the University of Cali, we were speculating that those microbes coming out of trichomes would be the microbes that populate the phylosphere or the root surface, because we have microbes that go all over these. And so emerging from trichomes would be the source of microbes that basically protect the leaves of the surfaces of the plant. So that could explain that. But anyways, that's that hypothesis.
Okay, we're going up to close to the 2 hours. So awesome. I don't know what else to say. This is a great show. Any final thoughts, Wendy, or any last? There'll always be questions, but we have work to do. Actually, I have a busy day ahead of myself. But thank you again, Dr. James White. And if any of your youngsters out there think about going to Rutgers, is there any other universities that are delving into these kinds of things in the United States, or do you have any? If someone's interested,
so there's endophyte work happening at a lot of universities in the United States, and some of them are kind of from the angle of plant pathology, using microbes to control disease. There's even I have a friend in Mexico, Guadalajara, Mexico, Miguel Beltran, that's working on this. So you might be able to go to Guadalajara, and do some work down there. I'd love to do that, really. Like I say, Cali, Colombia is a place. The Salisbury universities in Maryland. We got Mark Holland down there. There's some other places. I can't. All the places I can't remember. I mean, some of them are my friends, and I could. Bonnie Only. I think she's in North Carolina working on endophytes, but mostly for disease control. And, anyway, there are places, a little Googling around, and you can find those people who are working on endophytes, for whatever function you want, disease control, nutritional, to involve in plant breeding. It's becoming more and more popular area to work.
Did I hear you say that they're finding bacteria species inside of the trichomes?
Yeah, we have them in trichomes. A lot of plants have bacteria in the trichomes. So we have not per se. I do have a student working on hemp, but we have had a trouble getting material and getting permission to work on it. We're regulated very tightly by our university and our experiment station, so we can't go as fast as even the state can make it legal. So we have to go slower. But I'm trying to talk her into actually looking specifically, and she's been interested in the trichomes. It's just a little slow getting material going. And they go in trichomes in many, many different plants. You can see them there in many, many different plants. If you look in cotton, those cotton fibers are trichomes. They're loaded with bacteria in cotton. So you see in any plant, if you look closely enough, you stain the fibers on the fibers, the hairs, the trichomes on the leaves, for example, you'll see microbes in there.
Would that be something you could use that silver sulfate for?
You could use that silver sulfate. You could actually, yes. Silver sulfate should work. They should stain black inside the hair. So if you're going to do that, you can see them really good in hair. So you might be ahead of us then. If once you get silver sulfate, start looking at cannabis hairs. Would you let me know what you find?
Absolutely.
Yeah. Okay. Yeah.
Don't smoke it though.
Don't smoke it. No, don't smoke it. This is April Michi. Is my graduate student who I'm talking about. I'm not sure who the person was working on Metrolina, but April Michi from New Jersey. She's working on cannabis endophytes and using that.
Well, if she ever wants to come, or you ever want to come and do some studies in California, just hit me up. I got a big farm. We always have something going. So.
Oh yeah, next time you're trekking through this area, Crescent City, I'll show you around a little bit.
Okay, anyway, that's great.
I'm gonna wrap this up because I can just keep going and go. But, thank you so much, Dr. White, and thank you, Wendy. You saved the day. You're awesome, and look forward. I'm gonna keep following you. Is there any links? Do you have to get more of your work? Would you just go to Rutgers and just do a Google search?
Well, let's see. I have a Google Scholar page, so you can just look up the Google Scholar page. I have Rutgers. There's links at Rutgers. We have research pages at Rutgers. I have a lab page. So if you Google my name and Rutgers, that might come up. Or Google, I don't know, you can Google James White and endophytes and you'll get a lot of stuff too. So it'd be easy to find. A lot of the open accesses will come. I would suggest, there's hundreds of articles out there that I've written, 250 like that. But you don't need to see all those. I'd say go for the open access ones because that highlights things and otherwise you'll just be bored to tears. You don't want that. Give her a couple. You don't want to get all those. There's just too much detail there, and it's not all. There's some of them are on fungal endophytes, a lot on fungal endophytes, and I have collaborators all over, and we publish together, and so you'll see all that stuff. But I'd say the open access ones, those two that I recommended, are really good, and that ought to be enough. You can find the rest if you want, if you really want to see it.
Okay, good. I'm gonna wrap it up. I'm doing a quick plug tomorrow night on my show, regular time. I have somebody from Dr. Symes who's going to talk about their stuff, which is an interesting area too, with dealing with enzymes. But let's wrap this up. We got to do some work. Thank you guys so much for coming in. Thank you for watching. It was a great show. Had tons of good questions, and subscribe to the channel because it helps us put on quality content. This is amazing. So I'm gonna let you go back to work, Doctor. Are you going to be teaching in the classroom again later today? Are your schools back, by the way? Just curious. Are the kids back?
There are not many of them, but there are students taking. Some of them are back, and they're working online. They're doing their classes online. But I'm an editor for a few journals, and we've been working on a paper lately, and that's taken all my time. So all my editing work has backed up. So now I have to get back on that editing job and get articles that other people have submitted and get them sent out and all. There's no rest for the wicked, as they say. There's a lot of work that I have to do on those articles, and then I have to get back in the lab again. So it's constant stuff. But of course we love it. I love it. So it's not work at all, really. It's all fun and enjoyable. That's the benefit of my job.
I say that all the time. Being in the cannabis industry now is a fascinating. It's a fascinating field and a plant that people literally worship. So, having said that, let's go. I'm just going to close it down. Thanks again. Thanks for watching, and tune in tomorrow night for another show. Thank you so much.